Hi, my name's Thureya, and today I want to talk about the digestive system and how we get the nutrients from the food that we take into our bodies. So I'm just going to share my PowerPoint with you and then we'll get started. I'll make it a big screen. And then first of all, we're going to talk about some fun facts to do with the digestive system. So on average, a person produces 32 ounces of saliva a day. Some textbooks will say 1 to 1.5 litres of saliva per day, but thankfully we reabsorb most of it. Food takes approximately 7 seconds to travel all the way down the esophagus, which isn't long at all. The small intestine can stretch up to 23 feet, so it's referred to as small intestine because of the diameter, not the length. Your digestive tract is home to around 400 bacterial species. Your mucus lining of the stomach renews every 2 weeks, and digestion can even work against gravity. So that means if you're doing a handstand or something, you will still be able to digest your food. It will still travel down the esophagus towards the stomach against gravity, even if you're hanging upside down, which I don't recommend. Here is a diagram of the digestive system. So obviously we take food in via our oral cavity into the mouth first, and then there's two ways that we break down food. We break it down mechanically or physically using our teeth to start with. Our teeth and our tongue will manipulate the food, and we break it down chemically or enzymatically by the enzymes that are released from our salivary glands. So we'll take food in, our teeth will start chewing it all down, crushing it up into smaller pieces, and then our tongue, which is just a muscle really, will manipulate the food and push it towards the back of the throat. What we're trying to do is make the food smaller and soften it so it's easier to swallow. So once we've kind of manipulated the food and it's in a sort of soft little ball, we call that a bolus, B-O-L-U-S. And then we can swallow the food down at the back of the throat and into the esophagus. Now, the esophagus and the windpipe, or the trachea, which is the correct medical term for your windpipe, they run sort of parallel to each other. So there's a small piece of cartilage called the epiglottis, which kind of flaps over the top of the trachea, the top of the windpipe, to prevent food going down the wrong tube, so that it only goes down the esophagus. So if you've ever choked on a piece of food and you start coughing and spluttering, it's because it's went down the wrong way. So it should be going only down the esophagus, and thanks to the epiglottis, most people don't choke when they eat food. So then food will travel down the esophagus towards the stomach, and the stomach will then churn that food up. So there's lots of muscles that sort of crisscross in the stomach, and there's gastric juices that are released. The food will be sort of, again, chemically and physically sort of manipulated and churned with all the gastric juices and the motion of the muscles in the stomach. And then when the food is ready to leave the stomach and go towards the small intestine, be released into the first part of the small intestine, the food is then referred to as chyme, C-H-Y-M-E. So then the food will be sort of neutralized in the first part of the small intestine called the duodenum. Then it will travel to the middle part of the small intestine called the jejunum, where more digestion takes place. And then we're ready for absorption of that broken down food in the ileum. So the ileum, most of the absorption of all the nutrients, the amino acids, the glucose and things takes place in the ileum. Any sort of undigested food like fiber, things that we don't need, will be made into feces in the large intestine. So a lot of water reabsorption takes place here as well as there's bacteria in there which sort of help release vitamins from our food. And there's also bacteria, which anybody who's lactose intolerant, the bacteria will help break down lactose in the large intestine there. Then food travels towards any sort of undigested food. Feces travels towards the rectum and then will be released via the anus when we go to the toilet. So the digestive processes, the main ones I'm going to talk about, there's a few, but ingestion is when we first take food into the mouth. Propulsion is where we're moving the contents through the system. So we call it peristalsis. There's these wave-like muscular contractions that take place all along the digestive tract to help push and squeeze the food along. There's two main processes, like I said, mechanical breakdown. So in the mouth, it is with the teeth and the tongue, and chemical digestion by enzymes. But then when we get to the stomach, the mechanical breakdown is due to the stomach muscles churning and crisscrossing over each other and contracting the food. And then again, there's more enzymes in the stomach, again, helping break down the food. Absorption is where the digested food substances pass through the walls and then back into our blood circulation to be used by the body. And then elimination, any food that we've been unable to digest, like fibre and things, will be excreted when we make a bowel movement. So enzymes, what are they? They're biological catalysts which basically speed up the rate of reactions but remain unchanged at the end of the reaction. So digestive enzymes is the enzymes we're interested in here, and they help assist with the breakdown of food. So if we were just relied on physical or mechanical breakdown alone, we wouldn't be able to get all the nutrients from the food. We need enzymes to help break down the food further. So chemical breakdown is faster than mechanical breakdown because the enzymes, there's so many different enzymes that are helping do different things. We've got lipase enzymes breaking down the fats. We've got pepsin breaking down amino acids. We've got tons and tons of different enzymes, which I'm going to talk about further on the next PowerPoint. So enzymes work best at our body temperature. When we say an enzyme's working best, we say that it's at its optimum temperature and pH. So it's like the fastest that that enzyme can possibly work if it's in its correct environment. So there's some enzymes, for instance, in the stomach, it's quite an acidic area in there. There's hydrochloric acid in the stomach. So the stomach area is usually about pH 2. But by the time you get into the duodenum, where neutralisation takes place, it's a more alkaline environment. So the enzymes in the duodenum and the enzymes in the stomach obviously work in different pH environments. If you are to heat an enzyme up and boil it, it will basically become what we say is denatured. It loses its shape, and therefore it loses the shape of its active site, and it can no longer bind to its substrate to produce a reaction. So it's completely changed its chemical makeup. An example would be boiling an egg, and if you think about the white part of the egg becoming opaque, then basically you can't go back. Its chemical structure has completely changed and there's no going back the way. So some enzymes, like I said, might require a more alkaline environment, and some require a more acidic environment, especially the enzymes in the stomach. They won't work at their best if they're in the wrong environment, and sometimes they won't work at all if it's like, for instance, the temperature's been too high. The only acidic area is in the stomach, where the hydrochloric acid is. So here's the examples of the enzymes. Trypsin and chymotrypsin, they're made in the pancreas and help to digest proteins into peptides. We've got lipase enzymes, which digest triglycerides, which is our fats and our oils, to fatty acids and monoglycerides. We have amylase, which helps break down starch into maltose. That's the main enzyme which is in our salivary glands. Carboxypeptidase digests peptides to amino acids. Maltase helps digest maltose, a 2-carbon sugar, into glucose. A 2-unit of 2 6-carbon unit molecule. In fact, I'll maybe show you what this looks like if I just share my whiteboard. I can show you what I'm talking about. So say when we eat some starch. So starch is made up of lots and lots of glucose molecules joined together, like so. So I'll just actually write this up here. One glucose molecule has 6 carbons. 1, 2, 3, 4, 5, 6. So we call it a 6-carbon sugar. And then starch is just lots and lots of glucose molecules joined together in big polymer chains. So this was starch, it's hundreds of glucose units long. Here we've got our starch. So the main enzyme released from our salivary glands in the mouth is amylase. So amylase, just amylase, breaks down starch into maltose. Now maltose is just 2 sugar units, 2 6-carbon, I should say, 2 6-carbon sugars, 2 units. So it's 2 units of glucose makes maltose, is what I'm trying to say. So this is maltose. 1 unit of maltose here. There's another unit of maltose. So basically, amylase is breaking down the starch into smaller 2 sugar units. Then we've got another enzyme called maltase. So maltase, if I can move this up, maltase breaks down maltose into its single glucose units again. So in this case, we would be breaking down here, and then we'll be back to up here. We'll be back to this again. And then our body can use that glucose. We can use it straight away as energy, or we can store it in our liver and our skeletal muscles as glycogen, which again is just glycogen is big long chains of glucose molecules, but in the difference between starch and glycogen is more branched. There's branches coming off the big chains of glucose. So hopefully that is making sense. We go back to the PowerPoint now. So, lipase, amylase is what we were just talking about. Carboxypeptidase digests peptides to amino acids. Maltase digests maltose to glucose, which is what I just drew there. Sucrase digests sucrose to glucose and fructose. Lactase digests lactose to glucose and galactose. Peptidases digest peptides to amino acids. Maybe I should draw a little bit of the amino acids as well. I can move this up here. So if I'm talking about amino acids, what are amino acids? So the body can use 20 different types of amino acids. So we've got like valine, tyrosine, glutamine, etc. There's 20 different types. So amino acids, imagine this one's valine here, this one's tyrosine, and then I'll draw a glutamine over here. There's 20 different types. I'm not going to name them all just now. And then they all join together to make what we call polypeptides. Let me just draw these bigger like so. The red ones. And then I can make that thicker. Fine, go back to blue, make that thicker. So, maybe make a wee green as well. There's green. So we've got lots of different amino acids that we can have hundreds, even thousands of amino acids making these big, long polypeptide chains. And then initially we've got different enzymes which will break those chains, these big chains of hundreds of amino acids long, we'll break them into what we call peptides. So they're just shorter chains. So we're breaking them into short chains first, and then eventually we're breaking those chains down even further into their single amino acid units, so that we're just left with the amino acids on their own. So yellow one here, so that we can then use them, and then the body can then use that to build up another protein that the body might need. For instance, a hormone or another type of enzyme that the body might need. So again, we're breaking down any protein that we might need into these big long chains, into smaller chains first, and then into their individual amino acids. That's what we're trying to do. So we're breaking down large insoluble food particles into smaller monomer units that the body can then reuse again. So let's just go back to the PowerPoint and make this large. So ingestion happens in the mouth when we take in food. The mouth contains the teeth and the tongue to start the process of digestion by grinding and physically and mechanically breaking down that food into smaller units. And the salivary glands, we have 3 salivary glands: the parotid gland, the sublingual gland, and the submaxillary gland. So these glands are all releasing the main enzyme being amylase, which, like I said, breaks down starch into maltose. The submaxillary gland, or in this case, it also gets called submandibular gland, but I call it the submaxillary gland. It's easier to say. Basically, that releases the most. It's about 70% of the amylase that's released from these 3 salivary glands on either side of the jaw. It's the submaxillary gland that releases the most of it, 70% roughly. I think the parotid gland is about 20% and the rest is the sublingual gland. So these salivary glands are where all the amylase is being released, so that we're initially in the mouth alone to start off with, breaking down that food. We're sort of wetting the food and making it softer, and then it's going to be easier to swallow. Because imagine if you're eating crunchy, jaggy Doritos or something like that. If you're not making it all soft and easier to swallow, it's really going to hurt as you swallow towards the back of the throat, and then it's traveling down the esophagus. It would feel all jaggy and uncomfortable, I would imagine. So the effects of mechanical digestion are: food is easier to swallow. Like I said, we've made it into a nice soft bolus, it's called. And chewing produces a greater surface area for enzymes to act upon. So the enzymes are the part that are helping chemically digest the food. Physically is with the teeth and the tongue, chemically with the enzymes, and what we're doing is producing a larger surface area. What do I mean by that when I'm saying surface area? Let me just draw another example of that. So say that I'm eating a bit toast or a bit bread or something. So here is my bread or my toast. And the enzymes are acting on the surface area which is exposed. So they're going to act all around the surface of this bread. If I wasn't breaking it down, if I was just eating a whole loaf of bread in one go, the enzymes can only act on that outer area. But if I start breaking down that food and making it into smaller pieces using my teeth and my tongue, then we're breaking down the bread into much smaller particles and exposing surfaces that weren't exposed before. So we're creating a larger surface area. So now enzymes can act on all the inner surface areas which weren't exposed previously until we broke it down smaller. So now the enzymes can still act on the surface that was exposed, but also extra surface is now exposed thanks to us breaking it down into smaller pieces. So we have a larger surface area to work upon. That's what I mean by increasing the surface area. Let's just go back to the PowerPoint. So that was the effects of ingestion. So the main functions of the digestive system that I'm going to be talking about in this PowerPoint is digestion, the breaking down of large insoluble food molecules into small soluble molecules, which can enter the body cells into the bloodstream, and then our body can reuse them. And absorption, the passage of the small soluble molecules into the blood and lymph. So all the way through the alimentary canal, we have these 4 main layers. The lumen is this middle layer, this white space that you can see. This is where the food is going to pass. So the lumen is the bit that is the area that's exposed to the food. The next layer is the mucosa. This is a sort of mucus layer, which kind of protects the lining of the alimentary canal all the way through the tract. Then we've got a submucosa layer. This is made up of connective tissue. There's a blood supply in there. There's also glands and nerves. We've also got the muscularis, and there's 2 main muscles that are involved in making up this layer, circular and longitudinal muscles. So this muscularis layer is what makes peristalsis happen. Basically, food is able to be squeezed through the digestive tract because we have these 2 different muscles, circular and longitudinal, which are contracting behind the food and relaxing in front of the food to sort of squeeze the food down the tract. And then the serosa is at the outermost layer, all the way through the alimentary canal. So just to recap, the mucosa, this is the epithelial layer plus thin connective tissue and muscle. That was the bit that's in contact with the food. Epithelial cells are found all over our inner and outer cavities of our body. We've got the submucosal layer, the connective tissue layer, which also has a blood supply, a nerve supply, glands and things in there. The muscularis is the layer that I'm interested in when I'm talking about peristalsis. So those circular and longitudinal layers which contract behind the food and relax in front of the food to help push the food through the digestive tract. Because it's made up of smooth muscle, it's under autonomic control. So autonomic means we're not consciously thinking about digestion. We don't have to tell our digestive tract to start carrying out peristalsis and start digesting our food. It's all under the autonomic control of the nervous system. It's all automatic. We're not thinking about it. It happens without us having to be consciously aware that it's happening. And the serosa layer is the layer of epithelial and connective tissue which links the gut to the peritoneum, so the abdominal cavity. Here is a more detailed layer of the digestive tract. So you can see look up at the top of this here, basically. So the esophagus they all have the sort of same main layers with the mucosa, the submucosa, muscularis, and serosa. So you can see they've got the main layers. It's kind of when you get to this bit that touches the food, the layer that touches the food. So the esophagus, you can see, has sort of thicker projections. The stomach has these sort of gastric glands, these pits in the stomach, which I'll talk about when we go to talk about the stomach. The small intestine has longer finger-like projections called villi, and we'll look at the structure of the villi closer as we go. And the large intestine, again, has smaller little projections. So all the way through the tract, it's basically the same main 4 layers that I'm talking about. It's just the part that's really in touch with the food that is kind of different, which we'll see further along. So peristalsis, basically it's contraction of smooth muscle in the esophagus which pushes the food down towards the stomach. So like I said, they're like wave-like muscular contractions. So the muscles behind the food contract. Some people describe it as squeezing a tube of toothpaste. So you're contracting, pushing the toothpaste along. So the muscles behind are contracting and the muscles in front are relaxing. That makes sense, doesn't it? Because if the muscles contracted in the front and behind, the food just wouldn't move. It would just be blocked there. So basically you're pushing the food. Imagine squeezing a tube of toothpaste along the digestive tract. And the food, remember, is called a bolus. Once it's actually reached the back of the throat and traveling down the esophagus, we refer to the food as a bolus. So when we're first chewing that food in the mouth and manipulating the food with our tongue and pushing it towards the back of the throat, all of that is under voluntary control. We're aware that we're doing that, but by the time it reaches the back of the throat and then it's into the esophagus, that's all involuntary control. So here's a little example of this. So food starting higher up in the esophagus, you can see that the muscles are contracting behind the bolus. This little brown ball here is meant to represent the bolus of food being pushed down. As we move further down, you can see the muscles still contracting behind the food, relaxing in front, and the bolus is making its way down the esophagus towards the stomach. So the esophagus also gets referred to as the food pipe. It connects the throat to the stomach, and it's a long muscular tube that contracts to move food down and into the stomach area. The stomach is a muscular sac. They say it's about the size of 2 fists. It acts as a storage tank for your food. It contains hydrochloric acid and other chemicals that break down food particles. So the hydrochloric acid in there is released from cells called parietal cells in the stomach, and they basically, why do they release hydrochloric acid? Because the hydrochloric acid does 2 main things. It kind of kills any bacteria that we might have ingested with our food, and it also Well, I've just lost my train of thought there. It kills the bacteria, and it also, sorry, activates enzymes. Like, so enzymes obviously aren't active in our bodies all the time. We don't want them active all the time. So we have what we call precursor enzymes, where maybe it's an enzyme, but it needs a little bit cleaved off. Maybe a wee bit of protein needs cleaved off to make it active and expose its active site. So the hydrochloric acid helps activate inactive enzymes once food reaches the stomach. An example is pepsinogen, which is hydrochloric acid activates pepsinogen and forms pepsin, which helps to break down the proteins that we might ingest. So the stomach is made up of 3 different layers of muscle. They kind of crisscross over each other to help churn that food. The food is churned into what we call a semi-solid mass, which we refer to as chyme. I'll maybe just spell that out, shall I? Because it's not actually there, is it? So let me just go back to here and just say, so it's chyme. It's a CHYME. That's what we refer. So it was initially a bolus when it was in the esophagus. Once it's been churned and moved around, it's mechanically churned by the stomach muscles. So if this was our J-shaped stomach, we've got muscles that are sort of crisscrossing this way. We've got muscles that are crisscrossing that way, and then this way, etc. And all these muscles are sort of helping to churn that food, as well as enzymes helping to mechanically digest the food. I should say chemically digest the food, the enzymes. So let's go back to here. So food is churned to make a semi-solid mass, which we refer to as chyme. Then we've got the cardiac sphincter, which is at the top of the stomach, which basically opens to allow food to enter from the esophagus into the stomach. And then once the food's all been manipulated and churned around and gastric juices have helped break down the food further, the food will exit the stomach via the pyloric sphincter, which will open and then lead into the first part of the small intestine, which we refer to as the duodenum. So gastric glands in the stomach produce gastric juice made up of hydrochloric acid, which I've already mentioned. We've got mucus, which gives protection to the stomach lining from the acid. So why do we have that mucus there? We've got a big, thick layer of mucus to basically help the stomach not digest its own self, and also to protect the stomach from its from the acidic environment, because it's only a pH 2 in the stomach, so we're trying to protect the stomach from that acidic environment. And then obviously the hydrochloric acid helps activate the pepsin enzyme, so converts it from pepsinogen into pepsin to help break down proteins into smaller polypeptides, so that we can use the amino acids further down the line to maybe build up more hormones or proteins of some sort in the body. So the gastric glands in the stomach are found at the bottom of gastric pits, and the stomach is not smooth inside. It's not like all smooth and flat. It's highly folded. So let's just see if here's a sort of a stomach section. So you can see these high folds. So we're basically, this arrow's pointing down into the gastric pit, and at the bottom of the gastric pit is the gastric gland. And we've got a variety of different cells releasing different things. So we've got zymogenic cells or chief cells, which basically release different enzymes to help enzymes to help break down the food. We've got parietal cells which release hydrochloric acid. What else have we got? We've got the mucus cells, which are helping release mucus, which obviously makes sense. What else have we got? The zymogenic cells help release the pepsinogen, which gets converted into pepsin. This one's actually a better diagram, because you can see here's our stomach section here. We've got a little square here, and then it's zoomed in to help us see the gastric pits and then the gastric glands at the bottom. But if we go down here in all these coloured cells here, this is actually clearer. So enteroendocrine cells, or G cells, secrete the hormone gastrin, which is involved in gastric motility. We've got the chief or zymogenic cells, which secrete pepsinogen, which gets converted into pepsin. I was saying it was enzymes. It is an enzyme as well, gastric lipase. We've got parietal cells, which release the hydrochloric acid, and intrinsic factor, which is involved in vitamin B12 production. We've got mucus cells releasing mucus to help protect that surface lining. So basically, lots of different cells in these gastric glands, which are releasing lots of different substances to help in the process of digestion. So once all the food has been churned, I said it enters the first part of the small intestine, which we refer to as the duodenum. Now remember, in the stomach, it's highly acidic. So in the duodenum, the main process that's taking place is neutralisation. So we're trying to raise the pH of the food before it travels through the rest of the digestive tract. So basically, if we were to just allow that food, that chyme, to travel all the way through the tract at pH 2, two things would happen. You might damage the inner layer of the small intestine because of that acidic environment. The hydrochloric acid in the stomach is stronger than battery acid, they say. So you don't want that food traveling all the way through your digestive tract. And the stomach has a lot more mucus in it to protect itself from self-digestion. The duodenum and the rest of the small intestine are not as protected. So we need to raise that pH and make the chyme more alkaline so that it doesn't damage the rest of the digestive tract as it's traveling through. So two reasons is raising the pH, but also the enzymes that work throughout the small intestine can only work in more alkaline environments and not acidic environments. So we're neutralizing the chyme in the duodenum so that the rest of digestion and then absorption can take place. The next part of the small intestine is the jejunum. This is our middle section of the small intestine. And then the last part where most of the absorption takes place is in the ileum. And it's in the ileum where we find the villi, these small finger-like projections, which are helping to absorb the food back into the bloodstream. So here's a coloured diagram of the small intestine. You can see the duodenum is in this sort of blue colour here, leaving the stomach where neutralisation takes place, and we're raising the pH of the food in this area. The jejunum helps further with digestion, and then by the time we get to the ileum, we're ready to do most of the absorption is taking place in the ileum. So you can see in this diagram that the small intestine is highly folded. If we were to stretch that out, it would be about meters and meters long, 23 meters. Some textbooks will say even more than that, just kind of depending on the person. But it's, remember what I said at the start, it's to do with the diameter. The length of the small intestine is actually longer than the large intestine. The large intestine has a wider diameter, as you can see. And we'll talk about the large intestine later, but we're only interested in the small intestine for now. So digestion in the duodenum. The duodenum is obviously, I've said already, the first part of the small intestine, and it's the major site of digestion. Pancreatic juice containing bicarbonate. Bicarbonate is what's needed to raise the pH of that acidic chyme. So basically, bicarbonate is released from the pancreas. We've got bile released from the gallbladder, and it's all helping to neutralize the acid. Digestive enzymes are also produced in the pancreas. There's a variety of different enzymes: lipase, amylase, trypsin, chymotrypsin, like we saw earlier. These are all pancreatic enzymes. I spoke about the pancreas in my other video talking about the endocrine system because it's an endocrine gland as well. But in this case, we're not talking about the hormones released from the pancreas. We're only interested in the enzymes released from the pancreas to help with digestion. So you can see here, this is the bile, the gallbladder and the bile duct, the common bile duct. And then this part here is the pancreas. So both the gallbladder and this the bile duct leading off from the gallbladder and the pancreas have sort of ducts or vessels, little tubes and pathways that lead into the duodenum. So the pancreas is connected to the duodenum in order to release its enzymes and help digest the food further. And the gallbladder is releasing bile via the common bile duct into the duodenum to help emulsify fats. So lipase enzymes from the pancreas help break down fats as well into fatty acids and glycerol. Bile is also needed, not just the lipase enzyme, because it helps emulsify the fats. What do I mean by that? Let's just do another wee diagram here. Move this up. So if you've ever added oil to water, oil is obviously hydrophobic. So this is my fatty oil globule, say. So if I add that there, and then we add bile, I'll just depict that in green. So if we add bile, bile contains these little bile salts, which sort of separate the fats out. So what it's trying to do, because when oil kind of wants to come together, it's hydrophobic, so it's only attracted to itself. It wants to sort of gloop together in a big ball. When you start adding bile salts, you basically separate the fat globules away from each other, and the bile salts sort of surround the fat, the smaller molecules of fat, so that they don't all gloop back together again. So the bile salts are surrounding the fatty globules and preventing them from all coming back together again. So we call that emulsification. Or basically, the bile salts are helping emulsify the fats. Or a more simple term is we're helping break down fat. So that's basically what the bile salts are doing. They're helping separate the fat molecules and stopping them from all wanting to get back together again. So let's just make this big again. So we've got proteases released from the pancreas as well. What do you think they break down? Obviously proteins, polypeptides back into their smaller monomer units, which are amino acids. Trypsin is an example of a protease. We've got carbohydrates, which complete the digestion of carbohydrates back into smaller units of glucose. Amylase, maltase, sucrase. Notice a lot of the enzymes end in the suffix ase. So lipase, ase. Proteases, ase. We've got amylase, sucrase, maltase. A lot of enzymes end in the suffix ase. Not all, obviously, trypsin and chymotrypsin are exceptions, but the majority of enzymes, you know it's an enzyme because it ends with the letters ase. So neutralisation must occur in the duodenum, remember the first part, because there's not as much mucus in the duodenum as there is in the stomach, and so the walls of the duodenum aren't as protected. I already said that there's less mucus there than the stomach, and we don't want to damage any of the digestive tract. The enzymes of the duodenum don't work at an acidic pH. They need an alkaline environment to work. And finally, the contents of the meal have been reduced to the smallest soluble component parts and can now be absorbed into the bloodstream. So most of the absorption takes place in the ileum, the last part of the small digestive system. So if you were to slice through the ileum, you would see that it's highly convoluted. And these convolutions are called villi. So they're like these finger-like projections which help increase the surface area for absorption. The nutrients diffuse across the membrane into the bloodstream via the capillaries that are found inside the villi. So the main functions or benefits of the villi are to maximize diffusion into the bloodstream. The villi help provide a large surface area. They've got an excellent blood supply. They're moist to help the nutrients be more soluble, and they've got thin walls. You obviously want a thin barrier so that diffusion can happen rapidly, so that nutrient absorption can happen rapidly. So here's an example of what they actually look like in real life. There's the highly convoluted. If I was looking down the middle, the food would be in the middle part here. And then this is what I meant by finger-like projections. So what do I mean by increasing the surface area in this case? I showed you the example with the bread, but in this case, when we're talking about the ileum, the bit that touches the food, I said, is highly convoluted. So I'll try and redraw that picture that you just seen there. So it's highly convoluted, like so. So and let's just draw that as a circle. And so the food would be entering this bit in here. So say I was to slice through that, you can see that it would kind of look like this. If I was to slice and like stretch it out flat, it would look like this. Then if I was to pull from either end, stretch that out this side and stretch it out this side, these big villi, it would be very, 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 very long. And then so the enzymes can work. I'm saying enzymes, I mean, so food absorption can take place is what I should say. So food absorption can take place all along here. So we've got much more surface area to work on. If this small intestine wasn't convoluted like so, and it was just smooth, and the food was traveling in here, and if I was to cut through that, then I have much less surface to work on. It's so much smaller. There's nothing really to stretch out. It's just flat. Whereas if I stretch out these villi, I've got much more surface area for absorption to take place. I hope that's making sense. So moving on, here is just an example of one of those villi. So on the left-hand side, you can see in the center of one villi, we have what we call a lacteal. Lacteals absorb the products of fat digestion. So any sort of glycerol and fatty acids are absorbed into the lacteal, taken into the lymphatic system, and then transported around the body, eventually returning back into the bloodstream. The villi also contain a really vast capillary network around the lacteal here, and the capillaries that surround the lacteal help absorb glucose and amino acids back into the bloodstream to be used by the body at a later date. Surrounding the villi are epithelial cells, which also contain microvilli. So if we look on the right-hand side, we've got a thin layer, 1 cell thick, of epithelial cells, and there's microvilli there. Again, they're increasing the surface area for absorption to take place. So the epithelial cells that surround here are really a thin single layer. They contain microvilli to increase the surface area. There's also goblet cells which secrete mucus to help with solubility. So everything in the way that the villi is structured helps for absorption to be rapid, speedy, and quick, so that we can get the nutrients back into our system. So adaptations of the small intestine, which make it suitable for its function. It's several meters long, giving a large surface area for the absorption of digested food. The internal surface area is increased by thousands of these villi, those finger-like projections which we've just seen. The epithelial layer is very thin, 1 cell thick, moist to allow solubility, and it allows diffusion to happen at a rapid rate, because diffusion basically does not require energy, so we need a thin, moist surface to help facilitate diffusion. So the surface area is further increased by microvilli on the epithelial cells. And each villus has a network of capillaries which helps maintain the concentration gradient. What is a concentration gradient? It's basically the difference that exists between two areas of concentration. So molecules always want to move from a high area of concentration to a low area of concentration down a concentration gradient. And basically, maybe I should show you that. I always think of like a glass of orange juice or something, you know, when you've got diluting orange juice and you put the orange juice in the bottom of the glass. So say I was to draw a glass here. This is my glass. And then I add some concentrated orange juice, which needs diluted with water. And then when I add the water, eventually the juice particles will all sort of balance out, because molecules always want to move from a high area to a low area down a concentration gradient to balance out. So say this was 2 areas here, and I've got lots and lots of, I don't know, say I've got lots of glucose on this side. Say these are wee glucose molecules, but I've only got a few glucose molecules on this side of the concentration gradient. So these are 2 different concentration areas, and molecules are always going to want to move in this direction from the high area over here to the low area over this side to try and make everything balance out. So that's what I mean by diffusion. Molecules are moving from an area of high concentration to low concentration down a concentration gradient. So hopefully that helped. And I don't know if I mentioned that last point there. So we talked about the concentration gradient, and then inside each villus is the lacteal, which basically helps remove the products of fat digestion. So the last bit really to talk about is the large intestine. So it's mostly a structure known as the colon. The majority of absorption takes place in the small intestine, like we've seen, especially in the ileum. The large intestine's main job is to process any waste products and make it into feces to be removed by elimination from the body. So the main functions that take place in the large intestine are water reabsorption. And I said, I think at the beginning there, where bacteria will basically make use of any vitamins from the food there as well. And lactose, anybody who's lactose intolerant, there's bacteria in there to help digest any lactose sugar. So here is a coloured diagram of the large intestine. It's a much wider diameter than the small intestine. It has an ascending colon, a transverse colon, and then a descending colon is the last part leading out into the rectum and the anus. So the rectum is the final portion of the GI tract, the gastrointestinal tract. It connects the colon to the anus and it stores any feces there until it's ready to be eliminated. And the anus contains a ring of muscles. There's 2 sphincters there which open when we're ready to go to the toilet, which medical term we call that defecation. So lastly, I just want to mention the accessory organs. We've been talking about the digestive tract all the way from mouth to anus, but there are 3 accessory organs which help with digestion, and these are the liver, the gallbladder, and the pancreas. So while they're not strictly part of the digestive system, they are a massive help. They produce chemicals which aid digestion and help digestion to work properly, basically. So the liver, the pancreas, and the gallbladder, they're all found within the abdominal region. The liver is the 2nd largest organ in the body and it carries out a variety of jobs. So the largest organ in the body is our skin, the liver being the 2nd largest organ in the body. They say you can damage up to 75% of your liver and then it will still regenerate itself. So this bile that is stored in the gallbladder is actually produced in the liver. Bile is synthesized, it's made in the liver, but stored in the gallbladder. And bile is that dark green yellowy liquid which helps to emulsify the lipids, the fats. And it's produced in the liver but stored in the gallbladder. And the liver basically does just further processing of any digested food products. The gallbladder stores and releases the bile. The bile contains the bile salts which help emulsify those fats, separate out the fat globules so they don't all want to stick together, and it's released through the common bile duct into the duodenum to help break down those fats. So the correct word for breaking down fats is emulsify or emulsification. And the pancreas, like I said, it secretes hormones in blood glucose regulation. We looked at that in the video for the endocrine system. It releases 2 main hormones, insulin and glucagon, which help control blood glucose regulation. And if there's any problems with that, it can result in diabetes, type 1 and type 2. But in this case, in the digestive system case, we're just talking about the enzymes that are released from the pancreas. So amylase, trypsin, chymotrypsin, lipase, these enzymes are all released from the pancreas. And then there's little tubes or little ducts which release into the small intestine, the duodenum, the first part, to help break down any fats, carbohydrates, and proteins even further. The pancreas also, remember, makes bicarbonate to help raise the pH of the chyme that's just left the stomach and passed into the duodenum. So the bicarbonate raises the pH of the chyme. And that's it for the digestive system. So I really hope that that was helpful. Please let me know if there's anything If there's any questions, anything that I didn't explain properly or you'd like it explained further, and if you liked this video, please like and subscribe so that you'll get a notification when I make another one. I hope you enjoyed that.